To understand how the body moves, think of the nervous system as a two-part command chain. At the top are the upper motor neurons, and at the bottom are the lower motor neurons. Each has a distinct location, a distinct function, and a distinct clinical consequence when damaged. Lower motor neurons are large alpha motor neurons sitting in the ventral horns of the spinal cord, as well as in specific brainstem nuclei: the facial nucleus, the trigeminal motor nucleus, the nucleus ambiguus, and the hypoglossal nucleus. Their axons travel outward through spinal and cranial nerves to physically contact and control skeletal muscles. These neurons are called the final common pathway for all movement, both voluntary and reflex. That phrase is critical. It means no movement of any kind can occur without these neurons passing the signal to the muscle. Damage them, and all movement is lost, including reflexes. The resulting weakness is flaccid, meaning muscles go limp, tone is reduced, and deep tendon reflexes are diminished or completely absent. Above these sit the upper motor neurons, located in the precentral gyrus, the primary motor cortex, and in neighboring cortical regions surrounding it. These cortical neurons send axons downward to synapse onto lower motor neurons, relaying voluntary motor commands. Their axons form two major descending tracts. The corticospinal tract carries signals to the spinal cord, while the corticobulbar tract carries signals to the brainstem motor nuclei. Together, these are the highway for intentional, willed movement. UMN anatomy and function. UMNs include neurons of the corticospinal and corticobulbar tracts. They modulate voluntary movement and posture by synapsing on LMNs. LMN anatomy and function. LMNs begin in anterior horn cells, or cranial nerve motor nuclei, and extend to peripheral nerves and muscles, serving as the final common pathway for muscle activation. UMN lesion signs. Clinical features include spasticity, increased muscle tone, hyperreflexia, and pathologic reflexes, such as the Babinski sign. Acute lesions may initially cause hypotonia before hypertonia develops. LMN lesion signs characterized by decreased muscle tone and reflexes, muscle wasting, fasciculations, and flaccid paralysis. Reflexes are diminished or absent. Clinical relevance. Differentiating UMN from LMN patterns aids in localization and diagnosis of neurologic disorders, such as amyotrophic lateral sclerosis, affecting both spinal cord lesions, often mixed, and peripheral nerve or motor neuron diseases, LMN only. In summary, upper motor neurons, UMNs, originate in the motor cortex and brainstem, forming pathways like the corticospinal tract, and exert control over movement by synapsing on lower motor neurons, LMNs, in the brainstem or spinal cord. Lesions lead to weakness, spasticity, and hyperreflexia, with increased muscle tone developing gradually after the acute phase.